The sun had barely risen over Cambridge when James Watson burst into Francis Crick's office, his eyes wild with excitement. "I've got it, Francis," he exclaimed, waving a crumpled piece of paper. "The structure of DNA? It's a double helix." It was February 28, 1953, and this moment would mark the culmination of years of intense scientific pursuit. The race to uncover the secret of life had been fierce, with brilliant minds across the globe vying to solve the puzzle of DNA structure. Crick leaned forward, his curiosity piqued. "Show me," he said, reaching for the paper. As Watson spread out his rough sketches on the desk, the two men began to piece together the revolutionary model that would change the course of biology forever. Their journey had begun years earlier, in different corners of the world. Watson, an American biologist, had become fascinated with genetics during his studies at the University of Chicago. Crick, a British physicist, had turned his attention to biology after World War II, driven by a desire to understand the fundamental nature of life. Their paths converged at the Cavendish Laboratory in Cambridge, where they formed an unlikely but formidable partnership. Watson's biological insights complemented Crick's physical and mathematical expertise perfectly. As they worked tirelessly to unravel DNA's secrets, they were not alone in their quest. Across London, at King's College, Rosalind Franklin and Maurice Wilkins were making significant strides using X-ray crystallography to study DNA structure. Franklin, a brilliant physical chemist, had produced an X-ray diffraction image known as Photograph 51 that would prove crucial to solving the DNA puzzle. However, the competitive nature of scientific research and the gender biases of the time meant that her contributions would not be fully recognized until years later. Back in Cambridge, Watson and Crick's breakthrough came when they realized that DNA's four bases, adenine, thymine, guanine, and cytosine, paired up in specific ways. A always paired with T, and G always paired with C. This insight, combined with Franklin's X-ray data, led them to propose the double helix model. As they refined their model, using metal plates and wire to build a physical representation, the elegance of the structure became clear. The two strands of the helix wound around each other, held together by the base pairs, like the rungs of a twisted ladder. On April 25, 1953, their groundbreaking paper, Molecular Structure of Nucleic Acids, a structure for deoxyribose nucleic acid, was published in the journal Nature. In just over a page, Watson and Crick described the double helix structure and its implications for genetic replication. The scientific community was electrified. The discovery opened up new avenues of research and laid the foundation for modern molecular biology and genetics. It explained how genetic information could be stored, copied, and passed on from one generation to the next. As news of their discovery spread, Watson and Crick found themselves thrust into the spotlight.